All right, well, I'll be around. I'm going to kick off the next one here. I'm sure there'll be more questions. All right, so industrial hygiene is the science and art devoted to what we could say is the anticipation, recognition, evaluation, and control of those environmental factors or stresses arising from the workplace that may cause sickness or degradation of your well-being in one way, shape, or form, and that of the community. So I like this definition. It typically covers what we call CBRN. I put P on the end for physical, chemical, biological, radiological, nuclear, and physical. So heat stress, for example, is one of those that kind of bounces between physical and radiological, right? So these are different different ways of looking at this, but that's what we're talking about when we talk about developing an industrial hygiene monitoring plan, right? We have basically the whole world of potential contaminants is out there. What is it that you have? We have people that call all the time and say, I've got a problem. I've got a thing, something, and I got symptoms. Can you test the air? And it's like, man, I've got a lot of questions for you, right? Because we could just test the air, but that's kind of aimless and pointed in the dark, right? And what am I looking for? And so it all starts with designing the plan and anticipation. So that's why that first step there is anticipation. Do we actually anticipate that it's present? If we don't, then I'm not worried about it, right? In this room, am I going to test this room to make sure everybody's not exposed to methylene chloride? No, because nobody's using paint stripper in here and there's no other operations using methylene chloride. So I don't anticipate that it's present, so I don't worry about it, right? So we can eliminate a lot of things by right out of the gate. But sometimes it looks like this, where you see, it's pretty obvious. There's potentially some combustibility there, or here it's not obvious, but it is obvious because you just can't see, right? Because there's so much dust in the air because of a process that's pushing dust in the air. We know that there's problems. We know where the sources are. Sometimes it's obvious. Other times it looks like there's a source. Like this here was a project that had really large grain silica sand, and I'm going to use silica as some examples throughout this, but the particulate matter is really big and it doesn't fracture down. This is really interesting stuff. And before you know it, you realize that there's no problem here at all. Even though it's a big mess with this stuff all over the floor, it's not a problem. And so what is small? I mentioned that not being small enough. From a particulate perspective, if we're looking at that category, small is something that can get into your lower lungs, right? If it's something that your macrophages and the other processes in your lungs can't remove. So this is a shot blast media I took a picture of in a microscope with a reticle, but it's about the size of the smallest dot you can make on a paper in front of you with a ballpoint pen, and still see it. Like not a mash dot, but just the tiniest thing that you can still see. It's about this big, and this is 200 microns in diameter. We're looking at basically between 1 and 10 with a cut point of 4. That's a respirable particle. Okay, so that's right there in that sweet spot of going down deep in the lungs. You've probably seen this before, but there's if you calculate it out, I'm going to use silica again as that example. The PEL is 50 micrograms per cubic meter. 50 micrograms per cubic meter is the same as taking 1 gram, or like a sugar packet size of silica, and distributing it over an NFL football field with the end zones 13 feet high. Right? So that's really low concentration. But what are we trying to protect against? We're trying to protect against a lifetime of breathing this stuff in all the time, right? 45 years of a career breathing this in, and your lungs have no way of pushing it back out. So when it comes to particulate, we have to have really low levels for things that are respirable, so that we don't end up looking like this, right? With a lot of pneumoconioses and things like that. So the health effects can range. Obviously, we have it's going to make you cough and have problems with your inhalation and all that. You get down here to the small stuff, you have weird things like the cognitive delay and Alzheimer's, and this is just from particulate, right? So we do have some health effects that can range quite a bit, from CNS toxins like lead, mercury, arsenic, and other heavy metals, to sick building syndrome, headaches, eye irritation, things like that, all the way to everything else in between, speaking of methylene chloride. That's what we're doing here. So, but IH rule number 1, I like to say that be prepared for plans and education to take time and effort. That's where we put in a lot of our project time, is making sure that we have the right plan and the right approach to what we're trying to look for and what we're trying to rule out. Whether we're doing something that is like a clearance test, or something where we're trying to determine causality, or we're trying to determine compliance status, right? We need to make sure that we have a defensible set of data, and that starts at designing the study. So when we talk about creating a sampling plan, we have to think about that. Impacts and health effects. Let's talk about pneumo-ultramicroscopic silicovolcanoconiosis. Just kidding. All right. It's the longest English word, but it is actually an industrial hygiene word, though. So when addressing toxicological risk, compliance or controls, know the applicable reference values. If you don't know what you're going to compare it against, why do you even know what you're measuring? And if you don't know what you're measuring, what are you even doing here, right? So know what you're going to compare it against. And that might look like this, published standards, or it might look like stuff you pulled out of an SDS, or it might look like a NOAEL, a no observed adverse effect level, because you're working with some active pharmaceutical ingredient that is nowhere near getting a PEL, right? And so you got to figure out what's dangerous and then measure below that. You can also look around the environment at things that people are doing, like somewhere along the line, all these things are controls that were deemed necessary for whatever hazards present. That at the very outset, I'm talking at the very beginning when we're trying to determine if we anticipate a hazard, that's something you can walk in a room and say, big dust collector, what else is there in here? Where's that? Why is that here, right? I mean, if you're the first safety person in a generation at a facility and you walk in, maybe someone's thought about it before and there's indications of that and everyone's since forgot about it, right? So we see that too. So you can look at controls as a cue. IH rule number 3, IH observations and communications must contribute to guidance for business risk management, long-term planning, and workplace control expenditures. If you're not developing data as a part of your IH monitoring plan that's actionable or helping with decision making or helping to protect people or helping to determine compliance status or clearance or whatever, then again, what are you doing, right? It's just sampling for the sake of sampling. And so we don't want to do that. We want to make sure everything counts and everything has some good use to it. The scope and applicability. Let's talk about that. Basically, you want to make sure that your sampling plan covers all of the contaminants of interest, and you're not leaving anything out. But also, there are some ways that you can use analogs for a contaminant of interest when that contaminant is extremely hard to measure. For example, a food manufacturing facility might use an ATP test that's instantaneous read instead of taking 7 days to culture bacteria on a plate. And so they can tell right away if they have it clean or not. But when they measure ATP, they're not measuring bacteria, and that's what they want to remove. Well, it's okay to use an analog when that analog is reliable, right? And so there are ways to do that. Identify any regulatory-driven periodic sampling requirements. So like the silica standard, again, it has periodic sampling requirements. It has quarterly and biannual, and there's different thresholds that you have to meet along with a published timeline. Sampling methods, equipment calibration, sampling rates and duration. All of this stuff needs to be accounted for within your monitoring plan so that when you send someone into the field to do that, they know what they're doing, they're not confused, and you're not sitting out there with a pad and paper and a calculator trying to figure out what's next. That's problematic. All right, determine the number of samples, quality assurance, quality controls, sample handling and transport. Sometimes there's specific handling methods and temperatures you gotta hold stuff at, so make sure you're prepared for that. Document the sampling plan. Make sure that everything that you're doing is recorded so that it's actually defensible. Data analysis and reporting, again, you got all of your comparative analyses that you can use to determine what the outcome is, what the output of that observation is. And then review and revision. Review and revision is extremely important to make sure that, especially if you're on a recurring basis, that the next time that you do this, anything that you learned is applied to the future, and you can eliminate some of the potential problems. There are some specific kind of focus things like mapping. I'm going to leave this out because we're out of time, but this is basically what I'd say about this is when we do this, I spend a lot of time saying what it is and what it's not, right? So that people don't take this and run with it and come to the wrong conclusions. Because again, if you do all this work and you set it up right, but then the person looking at your data comes to the wrong conclusion because you led them that way, you're lost in the woods anyway, right? And so we've got to make sure people all the way through the end use of the data understand what the nuances of it are, what it can be used for, what it can't be used for. All right, I'm going to skip through this. I think you guys will have access to this, right, Tim? So there's some things in here to look at as well. There's also some interesting things to look at as we go forward in terms of wearable sensors and things that auto track and data log and things that maybe even measure things you didn't know you could measure, like using your heart rate to determine core body temperature, things like that, right? There's a lot of really interesting stuff out there that uses algorithms to calculate things. Are they the gold standard? Sometimes, sometimes not, but it might get you closer than you are. All right, I'll leave it there. Any questions about developing a sampling plan in 15 minutes?
Any questions for Dan on that
or on the heat stress? Yeah. All right. Thanks everybody.